This video is to do a quick and simple overview of the protein synthesis pathway as part of your Chapter 2, Module 2 OCR Biology A-Level specification. This video is focused for the OCR Biology specification, but it can be useful for other exam boards. Please subscribe to help my channel grow, and I'll continue to make videos covering the A-Level Biology specification. The main structures that are part of this pathway are the nucleus, the nuclear envelope, the rough endoplasmic reticulum, ribosomes, Golgi apparatus, vesicles, and cell membrane. When transcription has happened within the nucleus, proteins are synthesized in the ribosomes that are present on the rough endoplasmic reticulum. These proteins are packaged into vesicles from the rough endoplasmic reticulum cisternae. The vesicles move via the cytoskeleton to the Golgi apparatus. These vesicles fuse with the Golgi apparatus and the proteins enter the cis phase. These are modified and packaged again into vesicles that leave via the trans phase. The vesicles travel across the cells and fuse with the cell membranes for exocytosis to occur. The contents are then released. Any vesicles that are lysosomes that contain digestive enzymes remain within the cell to do their function. This is a basic overview of the protein synthesis pathway. I will go into more details when I make a video fully on protein synthesis, but for now, this is what you should know. Comment below anything that I can improve or anything that you want me to make a video about.